Globally, cardiovascular diseases are the leading cause of death. The World Health Organization says that in 2019, an estimated 17.9 million people died from these diseases. That's 32% of all deaths. The majority were due to heart attack and stroke. And as people are now living longer, often with multiple diseases, this will become more common.
There are many major hurdles in cardiobiology. For instance, the heart doesn't regenerate. So the heart is damaged, the damage is there forever. The heart cells are gone, they are gone forever.
There are therapies for heart disease, but how do doctors know which ones work, and how can new drugs be tested safely and effectively? That's where biomedical company Novoheart comes in. Owned by the Madeira Group for Drug Discovery, it's come up with a revolutionary solution bioengineered from real heart tissue. And here it is, a so-called heart in a jar. A mini living human heart pump, the first of its kind. They're grown from pluripotent stem cells, which can also be turned into other types of cells and tissues in the body. And they could be a game changer, helping to streamline and reduce the costs of testing medicines for heart disease. Professor Ronald Lee is the CEO and founder of Madeira.
Whenever a drug company wants to develop a new drug, so be prepared to spend about 10 to 12 years of time, several, let's say 2 to 4 billion US dollars, and yet the chance of being successful is less than 10%, or even some people would tell you even less than 5%. And the main reason is that everybody has been really using animals like rats, mice, you name it, for screening. And also whenever a drug fails, 50% of the chance it has to do with side effects of the human body.
Cardiac toxicity is a common serious effect of numerous new as well as marketed drugs, not just those for the heart. But Madeira's Novoheart is able to monitor the effectiveness and safety of drugs on several mini hearts in the lab at once, saving time. It also means no testing on animals or the patients themselves. All they need to create these heart constructs is a single drop of blood from a donor. How do you grow these hearts?
So we have figured out a way to instruct the stem cell to become human ventricular cardiomyocytes or human heart muscle cells. So within days, we can mass produce and generate billions and billions of cells. So you can see these heart cells as the fundamental building blocks. So once you have the building blocks, you can start assembling different tissues as well as a mini heart. And we do so by putting the cells into a specially designed bioreactor. So you can think of it as some kind of 3D printing, with the scaffolding, the extracellular matrices that the biologists call it. So allow a few days for the heart to grow and for the cells to come together, and they become a mini heart that is fully capable of injecting fluid.
Heart muscle cells, or cardiomyocytes, are unique because, unlike other muscle cells, they're highly resistant to fatigue, constantly contracting and relaxing, which controls the rhythmic beating of the heart. It takes 10 million heart muscle cells to manufacture a mini heart, which is just the left ventricle, one of the 4 chambers of the heart. A metal shaft acts like the aorta, the main blood vessel. When the mini heart pumps, it generates pressure and ejects fluid out of the metal shaft, which means researchers can measure pressure and look at the mechanical properties of the hearts. With this technology, they can make healthy hearts for screening toxicity as well as sick hearts for efficacy.
The reason why we're interested in the left ventricle is because it's basically the engine of the heart, and diseases that we're interested in, like myocardial infarction, heart attack, heart failure, they're basically left ventricular diseases.
Heart muscle cells have many applications. A cardiac tissue strip, like a fibre, can be manufactured with 1 million cells. And Novoheart is also developing heart patches to repair damaged cells.
But there's only a certain limit by which you can shrink your heart down to a critical size when you can still successfully model many of the human physiological parameters.
Nailing the miniaturization aspect of the lab hearts proved to be a very key turning point and came out of a collaboration with fellow University of California scientist Michelle Kine. Razor featured the biomedical engineer's creation inspired by shrinky-dinks, the flexible plastic sheets that shrink dramatically when heated but maintain shape and colour. Dr. Kine used the sheets to make microfluidic devices which can help diagnose medical problems using body fluids.
So we can pattern very inexpensively because we use Shrinky Dinks, which was a really popular children's toy when I was a kid. And so we pattern different materials onto the Shrinky Dinks. And if you remember back to those fun children's toys, you just pop them in an oven for 3 minutes and they come out. And instead of using them to make little jewelries or Christmas decorations, we actually take the active layer off. So we take the thin layer of metal or nanomaterials off and we put them into a soft stretchy material to allow us to get very high resolution monitoring.
So one very special features for the heart to function properly is the asymmetry. So for example, the electricity in the heart has to follow certain pathways in order for the heart to be able to properly contract. So that has to do with the symmetry. And that asymmetry is the result of cardiomyocytes or heart cells being aligned very systematically. And it turns out that her Shrinky Dink inspired technology allows us to be able to align cardiomyocytes or human heart cells in some very pre-designed systematic fashion. So that led to an engineered tissue design and also a series of studies, and with this tool, a very interesting tool.
For the first time, Professor Lee and his colleagues were able to see for themselves what effects several drugs had on a heart, albeit a mini one. In the past, antiarrhythmic drugs were mainly so-called blockbuster drugs, a one-size-fits-all drug earning a revenue of over $1 billion. With mini hearts, Novoheart is able to personalise antiarrhythmic therapy, guiding the development of drugs targeting specific problems. They can also develop hearts that carry human disease mutations and then trial therapeutics to return normal function. Dr. Roger Hajar is Madeira's Chief Medical Officer and CEO of Sadacor, a solely owned subsidiary of Madeira responsible for clinical trials. He's involved in the execution of clinical trials by choosing the most suitable therapeutics discovered using the mini heart and other platforms.
So you're not just treating disease before it becomes life-threatening, are you?
So we have multiple different platforms for treatments. Some, as you mentioned, are early on so that they don't progress to what we call end stage. But as we know, there are many patients at the end stage level from a cardiovascular perspective who need to have very specific treatment. So we also address that need, and it's a big unmet need. And gene therapy really comes into play in those settings where you have an end stage disease that you're trying to reverse back, and you need to reverse it at the molecular level.
In rare or inherited diseases, gene therapy has been used for many years. The therapy targets specific abnormalities, replacing the missing or defective gene in a diseased organ. To deliver gene therapy into the heart, a catheter is inserted through the arm or groin. The therapy product, in green, is carried to the heart by an inactivated virus, which means it cannot replicate and is injected into the coronary arteries. From there, these vectors carrying the genes are then distributed throughout the heart muscle, and they go into the heart cells and deliver their payload. That therapeutic payload is then distributed across the cardiac cell and translated into proteins. The proteins attach to the organelles or specialized structures that serve specific functions to keep a cell alive. So they improve the function of the cell and also the whole heart, turning a failing heart into a normal heart. Dr. Hajar says that so far there have been no safety issues with the delivery protocol for human patients. Now Madeira is focused on efficacy in multiple heart failure settings.
In another case, we can basically, instead of genetic mutations, we can basically train these hearts in a jar to become like what we see in humans, a very specific disease called heart failure with preserved ejection fraction, meaning they're very thick hearts that can't relax. And we can basically make these hearts in a jar to mimic that disease. And then we deploy our therapeutic and test and see, well, is it improving? And that would give us a direct view of what would happen in humans, because these are human hearts in a jar, and they're not, experimental models in animals, which sometimes do not directly correlate with what happens in humans.
Madeira has used healthy and sick mini hearts to discover new drugs and gene therapies and has US FDA approval to start a clinical trial. A number of other preclinical candidates are being fine-tuned to advance to the clinical stage. Madeira is also developing smaller sized tissues to increase the speed and efficiency of screening these clinical candidates.
Is this the future? Will this completely change healthcare?
For us, we've been working on the mini hearts, and my other colleagues are developing mini brains, mini liver, mini pancreas, blood vessels, you name it, right? So basically, by combining all these mini organs, we can make mini me, but except it's not like a walking individual, but it carries all the major critical organs. So human patients are not the guinea pigs anymore. And you can kind of like think about it as some kind of blood test. You go into the clinic, take a single draw of blood or some sort of tubes, and for testing this and that. And on top of that, the blood can be used for deriving these mini organs for testing a number of different things, and even the future of medicine. It's very exciting.